Bacterial cells do not divide by meiosis and mitosis. Instead, most bacteria grow asexually by a process known as binary fission. In this process, the bacterium copies its chromosome, elongates, and splits off into two daughter bacteria with a genetic makeup identical to the parental bacterium. Each daughter cell can continue the doubling process. This process happens rapidly, so the number of bacteria present in a solution can increase very quickly. When dormant bacteria are transferred to a fresh medium with plenty of nutrients, they spend some time switching their metabolic machinery from the dormant state to an actively growing state. The lag phase can last from less than an hour to days, depending on the species of bacteria. During the log phase, or exponential growth phase, the bacteria are actively undergoing binary fission. During the log phase, the bacteria double their numbers every generation period. The generation period can range from 20 minutes to days, depending on the species. As long as there are plenty of nutrients and little waste buildup, the bacteria will continue growing exponentially. It is convenient to represent the growth of the bacteria on a logarithmic graph, because the number of bacteria increase very rapidly with each subsequent generation. When the log of the number of cells is plotted versus the generation, the graph is linear. After the bacteria have grown for a while, and they are starting to deplete the nutrients in the growth medium, some of the cells begin to die. The stationary phase is reached when the number of new cells equals the number of cells that are dying. After the bacteria have depleted most of their nutrients and their waste buildup is getting too great, the bacteria begin to die in larger numbers than are made. If the death phase is carried on for a long time, most of the cells will die. If a cell is to survive, it must switch its metabolism back to a dormant state because there are not enough nutrients available for any more growth.
Now that you've seen those little videos, I just want to pull out a couple of the salient or important points to make sure you caught those in the video. Since you don't have a set of pages to take notes on, that's why we provided you with these last two. As you saw in that video, the growth of bacteria in a culture goes through is actually an exponential growth. As you can see on the graph here, when you graph the number of cells over time, it starts off pretty low here and then quickly accelerates up as you double the number in each of the successive generations. When you graph the number of the time or the generation number versus not just the whole number of cells but the logarithm of those, you get this straight line curve. So on this axis this is showing what is known as logarithmic growth, as you get a linear growth from over time with the number of cells. When we talk about being in log phase, we're talking about when cells are in this exponential growth phase where they are rapidly growing, and if you graph the log of it, you get a straight line. On this slide, I want to make sure we look at the various sections of a typical growth curve. You saw those on the video, but we want to give you a chance to jot them down and some of the details. The first phase, that flat part of the curve at the beginning here, right, that we saw, is, as you know, known as the lag phase. You'll remember during the lag phase, when you first inoculate a culture, the cells need to sort of start cranking out any enzymes they may need to digest material present in the media, and there's really not a lot of growth going on as they sort of get acclimated really to the media. In the next phase there's that exponential growth phase known as the log phase. That's what we just saw on the previous slide, where you have, again, we are graphing log of the number of bacteria on this axis, hence why we have this straight line growth in this area. During the log phase, you're doubling very, very rapidly. Again, with the typical E. coli at 37 degrees, every 20 minutes or so, you're going to get a new generation. The next phase, once a culture in this flat area up here in the curve, is known as the stationary phase. As you saw in the video, that's where the number of cells is not changing. Cells are still dividing, but also cells are dying at the same rate, so that you have no growth. So it's flat part of the curve. you're approaching, you're sort of at the carrying capacity for the culture that you're growing there. In the last piece of the growth curve, you have this exponential decrease in the number of cells that is known as the death phase. Here you have depleted the nutrients. The bugs have eaten everything. They're also wallowing in their own waste, in many cases which are acidic or poisonous, hence why they were waste products. And the number of cells are decreasing very, very rapidly, and that's where we've seen if we leave cultures in the incubator for too long, you get a lot of dead stuff accumulating at the bottom. So that's a quick overview of microbial growth, the factors that control growth, and how we will be able to take advantage of some of these things when we're working in the lab. Thanks a lot.